predictions. There are also wrappers that have been integrated to certain softwares, certain protein visualization softwares such as Camera X. So you can start a prediction directly from the program. It will not run locally on your computer. It will still connect with via internet, use collabs in the background, but you have this capacity to start a prediction directly from your visualization program. You also have some dedicated websites, and you can use also clusters. And in my lab, we use a lot the computing power of the Digital Research Alliance of Canada. The advantage here is that you can perform parallel computing, so you have also the capacity to model very large complexes thanks to high-end GPUs. And so you have a lot of flexibility in the way you have multiple programs and a lot of flexibility in how you can use these programs. And for those of you who are not already using AlphaFold, a good way to start is this collab, which is called AlphaFold. Very user-friendly. For those of you that have used the Jupyter Notebook, or Jupyter Lab, it's really similar. You basically have the code that can be hidden, and you define a couple of variables, and you can make your prediction this way. So how do these programs work? Typically, they work in one of two ways. Either they use multiple sequence alignment as a primary source of information. That's the way AlphaFold works, the way also AlphaFold works. Or they can use a large language model. In the case of large language model, the training was done not on protein structures, but on protein sequences. So using really practically billions of structures, you are able to learn something about the structure of proteins. So in those cases, all you need to do prediction is to have one sequence, your sequence of interest, and you will be able to obtain a predicted structure from this sequence. In the case of programs that necessitate a multiple sequence alignment, much of the power of these techniques rely in the use of this alignment. So even though you will provide only one sequence, you will need the program in the background will be looking for homologs of your protein of interest and generating these multiple sequence alignment. So this is the main program that we'll see. So the program that use multiple sequence alignment. So first, why do you need this multiple sequence alignment, and why does it procure information concerning structures? This is because throughout evolution, so your protein of interest is also present in related organisms, and there may be mutations happening here and there. And once you have a mutation at a certain region of your protein, you will typically have mutations occurring nearby in order to compensate for the change of residue at that particular position. So if we take the example over here, we have two residues that are close in space, but they're far away in the primary sequence of the protein. And what you see looking at these multiple sequence alignment, once you have a change at one position, you will have, so let's say we go from an arginine to a lysine residue, you will have another change that will occur at this other position, will change from an aspartic acid to a glutamic acid. So when you see those changes in the multiple sequence alignment, one of the reasons for those changes is a coevolution between two positions, and that informs you that those residues are likely to be close in space. So the programs such as AlphaFold, they use the power of this multiple sequence alignment to generate a structure, but they also generate a couple of metrics so that you can assess the quality of the predictions. In fact, there are two main quality indicators. One is called PLDDT values that inform you on the local quality of the structure. So this is given per residue, and it informs you whether the quality of the prediction at this particular location is very good, medium, or bad. So it's a scale between 0 and 100, with 100 being a really good prediction and 50 and below being pretty bad prediction. And typically residues which have low PLDDT values, they are typically intrinsically disordered, meaning that you don't expect at this position to have a secondary structure, a fixed secondary structure. So PLDDT values are one of the quality indicators that is generated during the process of generating your model. The other quality indicator is called PAEs, for predicted aligned error, and that informs you on the relative positioning of the residues. They're really useful to identify domains and their interaction. So basically, the information here is you have a graph of every residue of the protein on both the y-axis and the x-axis. And when you have low values, it means that if you know the position of a certain residue, how confident are you in the positioning of another residue? So if I go here from residue 25 to residue 50, I see that I have very low values, so dark green. It means that if I know the position of the first residue, I am very confident in the position of the second residue. That typically means those residues have a fixed distance in between, so they're part of the same domain or a same rigid body. So typically you will have green squares like this that will correspond to individual domains of your protein. If the individual domains are not interacting with each other, you will not see green at the intersection of these squares. But if they interact together, you will see those peaks in the diagonal position at the cross of those two squares. You will see other squares, and that means that those two domains, they interact with each other. And that's something that we use a lot in the lab because it informs us on which domains interact with which other domains. So not even looking at the structure per se, just looking at this quality indicator, we're able to see what are the domains that interact with which domains. Just to mention something that you may have already observed if you went through the AlphaFold database and look at a couple of protein structures. They practically all have this compact shape. So here I took the example of a big protein, BRCA1. So it's about 2,000 residues. And you see that there's a lot of residues, and it's all compact in a kind of big ball of spaghettis. That is somehow an artifact of AlphaFold. all the structures will have this compact appearance. It may not reflect the real state of your protein. And that's one of the uses of the PA diagram. It informs you, can you trust what you see? Can you trust the position of all these residues? And when I look at the PA here for this BRCA1 protein, what I'm able to say is that there are two domains, maybe a small secondary structure in the middle of the protein, but I cannot say anything about how most of the protein will appear. So I'm not able to say if the protein will be more elongated, if it will be a compact protein. That's something that you can tell from looking at the PA. Now, one of the big applications of AlphaFold is predicting interactions. And practically from the start, AlphaFold was used by different groups to predict those protein-protein interactions. So at the beginning, it was frequently done by just inserting glycine linker between the individual proteins. Now this has been refined. You can submit to AlphaFold different proteins, and it will generate prediction for the complex. But in some cases, for example, ESM-Fold, you still don't have the capacity to input multiple protein sequence. You can still use this trick of using glycine linkers in between your proteins. And when you do this, you can see that you can generate very decent protein complexes. So it goes better and better with newer versions of AlphaFold and of all these different programs. But if you have here, this is a protein that we work with in the lab, and you have both the experimental structure of the complex and the predictions, and they're closely matching. So what you do to generate those structures of protein complexes? In fact, what AlphaFold will do is it will pair your protein sequences. So it will put one of your protein you will have one protein, and immediately after that you will have your second protein. And AlphaFold will look in different species what is the closest protein for each of your protein of interest, and it will try to find coevolution patterns in between your two proteins of interest. So it's basically the same idea as I presented. From the coevolution of protein residues, you get insight into the protein structure. So by having here multiple sequences of both proteins coming from different organisms, and each line corresponds to one particular organism, you will get information on how those two proteins interact together. So this can be conveyed in a certain graph. AlphaFold typically generates graph like this that lets you see how much sequences were found to generate the multiple sequence alignment. Typically, for AlphaFold to give you good structures, you typically need at least 30 sequences. But you need those sequences to be somehow different from each other. If you have 30 sequences that are really similar to each other, you will not have a lot of information to deduce those coevolution patterns. But if you have 30 sequences that all correspond to homologs of your protein, but that explore very, very different protein residues, then you will have a lot of information to generate a good structure in the end. So what we do in the lab, we typically do screens for protein-protein interactions. And one of the way we're using to detect that we have indeed an interaction between our proteins of interest is looking at this PAE diagram. So if we do this, if we do the prediction for a complex between two proteins, AlphaFold will generate a PAE diagram very similar to the one that would be generated if you have only one protein, except that here you can look at those quadrants, those two quadrants that inform you whether you have an interaction between your first protein and your second protein. So in the lab, we've developed script to automatically look at those regions of the PAE, and that's a way in which we can make prediction for a lot of potential protein-protein interactions and quickly obtain this data and know what are the proteins that interact with each other. So just as an example from my lab, so we have the scripts that will look at the PAE for different regions for potential interactions, and we can sum up all of these information in really small tables that allows us to say what are the potential interactors with one or another region of the protein. Then we can look at the complete PAE, see where this interaction occurs, and then we can have a look at the structure, and then we have a judgment call to say whether this looks real to us or if it may be a less good of a prediction. Now I will go towards certain things that we see in the structures that are generated by AlphaFold. So for those of you who have not used a lot AlphaFold, you may be surprised by what types of prediction you obtain from AlphaFold. AlphaFold will typically return the structure of your protein of interest as if it was part of the biggest complex as possible. So in other words, it will not generate the structure of your protein as if you were expressing it, for example, in bacteria with none of its usual protein partners. It will typically generate the configuration as if it was present with all of its protein partners. So it will generate typically the structure of the most complex state of your protein. So, for example, if you go into the AlphaFold database and you look for this particular protein, you may be intrigued by the structure that you see. You see a couple of secondary structures that seems to be hanging all by themselves. It's not something that you would usually see in a protein that is alone. But in fact, this protein is part of a really larger complex, the ribosome, and the prediction that AlphaFold generated, is in fact the structure of this protein as if it was part of a complete ribosome. So this is something that we have to consider when looking at predictions. They're not only predictions of what the protein looks like by itself, but what it could be if all of its protein partners were present. That's something that we look for a lot in the lab. So when, for example, we see a prediction and we see isolated secondary structures for our protein of interest. So for example, here we have two beta strands that seems to be hanging out by themselves. They're probably not like this in the protein when the protein is alone. This is probably a sign that this particular part of the protein interacts with something. So when you see those things in the structure of your protein of interest, it's a sign that maybe you should be looking for something. Maybe first look if a similar structure exists in the PDB, and we'll see tools for this in a couple of slides. You can try to generate an oligomer of your protein. Sometimes protein will form homodimers, homotrimers, or other forms of oligomers. Or you can, or you could screen for potential interactors. One thing that comes frequently when people start playing with AlphaFold is, can we use this for predicting the effects of mutation? And here I have to urge you to be cautious about what you do in predicting the effects of mutations. Sometimes you will see that it fits a model that you have for the effect of the mutation. But you can also have cases like this. This is one of the proteins that we work with in the lab. And if we start making mutation in the hydrophobic core of the protein, we can mutate a lot of those residues. And AlphaFold or other programs will still generate overall practically the same structure. Even though if you were to make these mutations to the proteins in vivo, there is no way that such a mutated protein could fold into the same, that you would not perturb the folding of your protein. But in the prediction, especially if you remove part of the residues, so for example, if you go from an isoleucine to an alanine, it's possible that the structure that will be generated will be very similar to your wild-type protein. And one of the reasons for this is, especially in programs such as AlphaFold that use multiple sequence alignment, what you mutate is just one line of your alignment. But when AlphaFold will search for homologs, it will see that at this particular position, most of the proteins have other residues. They have hydrophobic residues, for example. And this somehow, the residue that you've introduced as a mutation will get pushed over, and AlphaFold will see what's happening in the other proteins, and the model you will get at the end will correspond to the native protein. So always remember, AlphaFold and most of these programs were trained not on mutated proteins, but on the wild-type proteins. So you have to be cautious. Some people, some groups have experimented with this. Can we use AlphaFold to predict mutations? In certain cases, under strict conditions, maybe you can predict the effects, but in a lot of cases, you have to be prudent with what you do. But that doesn't prevent you from looking at the structure of the wild-type protein and speculating on the potential effects of mutations. One thing that AlphaFold would not do is model residues other than protein residues. It will not model nucleic acids. It will not model ligands, cofactor, or post-translational modifications. But so this is an intrinsic limitation for now of AlphaFold. But what's interesting is AlphaFold will predict the complex state of proteins. So frequently, if your protein of interest is binding a certain ligand, you will have the space available in the protein, and the protein will be modeled in the ligand binding state. So you could dock your ligand of interest into those structures. Or another way is using this program, which is called AlphaFold, that will look at similar structures in the PDB. So they will look for proteins that have a certain degree of sequence similarity with your protein and see, are there ligands that are bound by these proteins in the PDB, and you can transpose those ligands to your protein of interest. So this is something useful to know. So it's very easy to use this software. You just have to enter either a UniProt ID or your structure of interest. It will look for proteins that have a certain degree of sequence identity with your protein of interest, see what are the different compounds bound by these proteins, and allow you to transpose these ligands. Other things that are useful to know when using AlphaFold, you can do structure comparison and have a little more idea where do these structures come from. Are there similar structures that exist, either other models or experimental structure? So there are two programs that we use a lot to perform comparison. One is called DALI. It's an exhaustive search, 3D search. It takes time, but it typically generates very good results. Another software that you can use is called FoldSeek, and this one is much quicker, and you can perform ultra-fast structure comparison either between your protein of interest, the structure of your protein of interest, and the PDB or other models generated by AlphaFold. So now we'll just go very quickly through different projects, mostly collaboration projects, just so that you have other ideas of what you can do with AlphaFold or other programs of structure prediction. So the first one is actually a research on rare diseases that is led by a colleague of mine, Marc Lucier. So he's interested in a protein which is called RNF13, a ubiquitin E3 ligase. And what he knew at the beginning of this study is that a mutation in a dileucine motif is the cause for a disease which is called developmental and epileptic encephalopathy. So it's a disease that starts very young and very devastating. And he was looking to understand why this mutation at this particular localization would cause such an heavy phenotype. So what we did is we looked for interactors of the NF13 protein, and were able to find one protein, AP3S1. So it's a member of a protein which is involved in the endolysosomal transport of proteins. And what's interesting is that in this protein complex, we have those two leucine residues that of RNF13 that are interacting with this protein AP3S1. So the structure that we obtained, it explains beautifully the conservation of those residues in AP3, in RNF13. And the group of Marc Lucier could do experiment to confirm that this is a real interaction and that indeed it affects the localization of the proteins. So it's a case where obtaining the structure by large-scale protein screening was informative and was helping in getting more information about the biology of this protein of interest. Then we can go further. We can cross this information with other databases, for example, post-translational modifications, and then we can start refining our vision of the biology of this protein. So we know, for example, that there is a potential phosphorylation site in this threonine residue. And so it can guide us in understanding, okay, maybe there is a regulation of this interaction by phosphorylation, and that's something that will be tested in the near future. Another example of using this information, the models that are generated by AlphaFold, it's another collaboration with another colleague, Lecas-Leno at UQAM. So the group of Lecas-Leno does a lot of proteomics and metabolomics. And in this case, we'll not give too much details, but they have certain compounds that can react with proteins. So they know that this particular compound can react with cysteine residues. And here, the interest of using protein structures is that we're able to capture the three-dimensional environment in the proximity of those residues that are modified by this compound. So this allows us to understand, is there something special in 3D that promote the modification of the proteins at a certain residues? We can look at what are the residues that are around this cysteine residue that will be modified. And we can ask the question whether those residues that are present in the vicinity, would you expect them to be in proximity of a cysteine residue? And if you have a major change, it means that maybe those residues contribute to the specificity of interaction with this particular compound. So as a conclusion, structures that are generated by AlphaFold or the other programs are really useful to explain or contextualize experimental results, and they're an excellent source of inspiration to generate new hypotheses. So we typically launch big screen in the labs, and we're able to generate those hypotheses knowing with which protein our protein of interest is interacting. They're excellent to predict the structure of monomeric proteins, excellent to define domain boundaries, very good at predicting protein-protein interactions, and to identify disordered regions. Of course, there's some limitations also, still emerging capacity to predict protein stability and dynamics. Sometimes it can have difficulty obtaining or generating a model of alternative conformations. So some proteins, it is known that they exist in multiple configurations, and frequently AlphaFold will be biased for one of these conformations. Still have a limited capacity to deal with post-translational modification, mutations, or ligands. And we know that for certain interactions, the models are likely to not to be really good as for the rest of the protein. So for example, antibody-antigen interaction, it's still, the quality of the prediction is still pretty low compared to other forms of predictions. And finally, there is a limited capacity to influence the prediction with either experimental or environmental conditions. So for example, you wouldn't be able to screen for the effect of pH on the structure, on your structure of interest directly using AlphaFold. So thank you so much once again for the invitation, and I would be happy to take questions.
Thank you very much for your presentation again, and we can start with questions from our audience. Otherwise, I'm going to ask questions. Okay. We have multiple people with questions. Okay, we can start. I saw Zevi has a question.
Yeah. Hi, Laurent. Thank you so much for your talk. It was a really great overview of AlphaFold. Really liked it. Thank you so much. Um, so at the very end there, I saw that you mentioned um, condensets, and that's something that's of particular interest for my lab. And so I was interested in knowing, are there any efforts to combine AlphaFold and its capacity to identify PPIs and identify disordered regions with physics-based or molecular dynamics-based software to sort of maybe predict how disordered regions would act, and how what their dynamics would be within the context of a structure. I was curious to know.
So you're asking about how we can obtain more information about the dynamics of the protein using AlphaFold or a combination of AlphaFold and other software. Am I getting your question
Yeah,
right?
yeah, exactly.
Yeah, there's more and more people are doing molecular dynamics after generating models with AlphaFold. You have also a couple of strategies in order to increase the diversity of structures that you would be looking at. So dynamic state of the proteins. And one of the way to do this is I will share the screen once again. I put here for alternative conformations, but it really works for dynamics. The idea is that you will decrease the size of the multiple sequence alignment. So this was done for a protein that can exist in two different conformation. And what they show is that if you use a really large set of a really large multiple sequence alignment, you will tend to bias, you will tend to converge towards one particular configuration. Whereas if you only select a few sequence, you will generate more conformations. And there you were able to explore more conformations of your protein. So people tend to use a combination of both generating a larger diversity of models and performing molecular dynamics or normal mode and changes on the protein just to assess the landscape of configuration of dynamics.
Cool. Thank you so much. I had a second question, actually, if that's not a problem. You had mentioned before about predicting protein-protein interactions proteome-wide, and I personally work with yeast, and so I was curious to know. You had mentioned an effort by David Baker in the past. But if I remember correctly, that was with a more restricted set of proteins. I was wondering, how close are we to like proteome-wide prediction of protein complexes and binary PPIs?
To my knowledge, it's always guided by, for individual proteins, you can do the screen against practically all the proteome. That's doable, but all the proteins has, to my knowledge, not been done. But people will use constraints in terms of what is maybe possibly already known in terms of protein-protein interactions, or sometimes in the lab what we would do is do a restriction based on subcellular localization. So if we know for a particular compartment that may not be either the nucleus or the cytoplasm where you have a lot of proteins, but if you're more restricted, let's say you're in the lysosomes, then it's more doable to do an all by all matrix. But complete for the cells, I have not seen it.
I see. Thanks. And I'm sorry to drag on like this, but this is of personal interest to me. But if you work with a data set of say 10,000 or 20,000 PPIs that have some form of evidence that they interact, is that like a reasonable number in terms of is that a reasonable number for predictions with AlphaFold or other software?
The reasonable numbers for us is in the 1,000 of proteins. But that all depends on what you have access to in terms of computing capacity. Using the resources from the Digital Alliance of Canada, that's typically what we have using our present allocation. You could probably, with the default allocation, you're probably okay up to I would say in the thousands. If you have more generous allocation, then you can go 10 or maybe 100,000.
Okay, thank you. That's very helpful. Thank you so much.
My pleasure.
Do we have other questions, or I'm going to ask some questions to you? Okay, I don't hear anything. So my question is, for the time scale wise, what can be done to make things faster?
To make things faster, you have the capacity. I mean, AlphaFold has when you're doing a prediction with AlphaFold, it will typically do cycles of prediction, and then the word is recycling. So at one point, you will be looping and practically improving on previous predictions and continue improving on the predictions. So you can reduce this recycling number and you will be able to obtain faster predictions. So typically, at the beginning, AlphaFold was using a recycle of 3. Then it was suggested that if you would go to higher numbers, you would get better model quality. So I think that for CollabFold, it goes, from memory, it's up to 20. But if you reduce this number, you will get quicker predictions. The other way is that, in fact, there's not only one model that was trained by AlphaFold. There's 5 models that was individually trained. And that's why at the end of generating a structure, you get 5 models typically. If you use only one of these trained models, then you would be able to divide by 5 the amount of time you need to do the predictions. You will only get one model, but you will be 5 times faster. Other than that, typically we don't use the relaxation at the end of AlphaFold runs. So this is a step of energy minimization that is performed at the end. We typically find that it's we prefer to see the results, and then if the results are interesting, then we will do our set of minimization. But this only slows the pathway. So we typically don't minimize unless we're confident that it will be an interesting structure. So basically, we generate the model. We look at the PAE. If the PAE is interesting, then we go ahead and minimize, but otherwise we don't.
Okay.
Sorry.
Yep. Yeah. I have one more question. That's why. It's personal interest. Is there so in the AlphaFold database, I know that the structures are available in the PDB format. Is there a way, or is it available? The generated multiple sequence alignments, are they reachable for each protein, or they're only sharing the structures in the database?
To my knowledge, it's only the structure. But generating if you were to generate for example, the human proteome, that's something we've done. And with MMSEX2, it only takes a day. Okay. It's not that it's not that long.
Okay. I know that AlphaFold is using MMSEC2, and that's why it's much faster compared to the AlphaFold multimer prediction alone. Okay, so these were my questions. If you don't have more questions, we can say thank you again. Okay, I don't see any more questions. Thank you so much again for the talk. It was very interesting. Thanks a lot.
Thank you so much for the invitation. It was a pleasure.
It's our pleasure. Thank you. Bye-bye.
Goodbye.